Good morning, students. Myself, Dr. Mrs. Damini Motwani, Assistant Professor, Dada Ramchand Bakru, Sindhu Mahavidyalaya, Panchpavli, Nagpur. Today I am going to talk on the topic Bacterial Growth Curve. Bacterial growth curve is the study of growth of bacteria in a liquid nutrient medium. We will study how a bacteria divides, how a single bacterial cell divides into two by a process known as binary fission. Now this bacterial growth curve, it is basically divided into four phases. These four phases are studied by drawing a curve. We take x. On x-axis we plot number of cells and on y-axis we plot time. By plotting number of cells on x-axis and time on y-axis, we get such type of curve. Now this curve, bacterial growth curve, it is basically divided into four phases. First is lag phase, second is log phase, third one is stationary phase, and fourth one is death phase. So you should remember these four phases: lag phase, LAG, PHASE, lag phase. Second one is log phase. Third one is stationary phase, and fourth one is death phase. This is lag phase, where there is no increase in the number of cells. This is log phase, where there is a exponential increase in the number of cells. This is stationary phase, where there is again there is a balance between the number of cells which are being reproduced and the number of cells which are being dying. And fourth one is the death phase. Now, in detail, we will see what is the difference between these four phases when we are studying the bacterial growth curve. So, lag phase, as the name specify, in lag phase, the cells are not at all dividing. The bacterial cells are not dividing. Only they are preparing themselves for the division. They are increasing in size, but they are not dividing. The copy number of the cells is not at all increasing in the lag phase. So there is no increase in the number of cells in the lag phase. Second phase is the log phase, which is also known as exponential phase. In this phase, the cell number increases exponentially. Exponentially means 2 to the power n. N represent the number of generations. For example, first the cell has been divided from 1 cell, 2 cells are produced, from 2 cells, 4 cells are produced, from 4 cells, 8 cells are produced, from 8 cells, 16 cells. In this way, there is an exponential increase in the number of cells during the log phase. During this phase, each and every bacteria which is being dividing, they utilize the nutrients which is present in the liquid medium. Means they are exhausting the nutrient medium. The third phase is known as stationary. Stationary means balance. Now in balance in what sense? Balance in means here is the number of cells which are being dividing and the copy number which is being increasing. It is basically not increasing because, for example, if 10 cells are being increased, the 10 cells also get die. So there is a balance. The cell number, though there is the presence of division in this phase, but still the copy number means there is no increase in the copy number of the cells because the cells being are dividing, the exactly same number of cells are again being die. So why there is a die? Because in the log phase, the cells which has been divided during division, they are utilizing the nutrients which is present in the medium, as well as they are producing the waste products which are toxic. Means the waste product due to which there is a cells die in the stationary phase. So there is a balance. The last phase is the death phase. The death phase, because the number of nutrients or the amount of nutrients which was present in the medium has been exhausted in the stationary phase, and now there is no more nutrient available for the bacteria. So the bacteria is not able to divide. So there is no division, but the cells start getting die, and hence there is a decrease in the number of cells in the last phase, which is known as death phase. So these are the four basic phases of a bacterial growth curve. Lag phase, where there is no division, the bacteria only prepare themselves for the division. Log phase, where there is exponential increase in the number of the cells. Stationary phase, where there is a balance between the number of cells reproducing and the number of cells getting die. And the last phase, there is a decrease in the number of the cells. It is being said that the log phase is reciprocal to the death phase, means the second phase. As there is an exponential increase in the log phase, exactly there is an exponential decrease in the number of cells in the death phase due to utilization of all nutrients and production of waste products. Thank you.